As you can see, the eggs laid in the wheelbarrow have developed. In fact, we can see that this organism has a mouth and a body plan because it is chewing on this petal. Just like vertebrates, invertebrates like this go through the stages of early cleavages, then gastrulation and organogenesis. Unlike the vertebrates that we're studying, the cleavages that this embryo goes through during early morphogenesis do not involve the making of cell membranes, but just copying of nucleuses. Gastrulation is the same. Also, unlike the vertebrates we're currently studying, this mosquito larvae will activate genes that cause body plan development twice, because it will go through 2 stages of organogenesis. This is a larva. It is a mosquito larva. After it finishes its life feeding this way, it will pupate, and then it will go through organogenesis again to become the blood-sucking flying organism we all know and love, the mosquito. This egg is super early in development. I really hope that you can all see the very small embryo. It has a beating heart. If we were in class, you'd be able to see this. It's right by my thumb.
Cytoplasmic determinants are unevenly distributed chemicals in a developing embryo that affect gene expression and epigenetic inheritance. The establishment of the dorsal ventral axis occurs at different times in different vertebrates. It happens at fertilization in frogs and salamanders. It happens at around the 16-cell stage in mammals. In the amphibian, sperm fusion initiates pigmentation changes in the fertilized egg. The gray crescent forms opposite the site of fusion. The gray crescent marks the cells that will, in subsequent cleavages, express the gene Noggin. These cells will form the dorsal side of the animal because Noggin represses BMP4, another gene product, and BMP4 causes ventral identity. In addition, Noggin expression will initiate the dorsal lip of the blastopore further along in development. The dorsal lip of the blastopore, as it moves from anterior to posterior during gastrulation, will direct the identity of cells moving through it genetically. Whether we examine an amphibian, a mammal, or a bird, there's very little change in size for the embryo from fertilization until gastrulation. During organogenesis, the genetic identity of cells has become more restricted, and there's some level of epigenetic inheritance from parent to daughter cell. In addition to this, more genes are activated. The embryo begins to grow, and genes begin to initiate the development of transient structures whose fate in the adult body plan is now known. So even at the very beginning of organogenesis, when our embryo is just this little tiny developing organism sitting on this blastodisc on the yolk, major gene expression events are initiating the construction of an entire bird body plan. So that by the time the embryo gets to this stage here, where we can see its beating heart by candling the egg, a ton of really awesome things have already happened genetically and as far as tissue migration and structure formation is concerned. So now let's connect the expression of genes to development. So it turns out the pigmented side of the embryo called the gray crescent is going to become the dorsal lip of the blastopore, or some small part of it will, and both of these regions then are known as what's called the Spemon-Mangold organizer. And one of the genes responsible for the Spemon-Mangold organizer identity is the gene called Noggin. Noggin expression is necessary for the Spemon-Mangold organizer to do its thing. So sometime during early morphogenesis, cells on what will now be the dorsal side of the embryo express the Noggin gene. Expression of the Noggin gene inhibits the expression of BMP4. BMP4 causes ventral identity and its inhibition causes dorsal identity. The embryo now has a ventral dorsal axis. So the dorsal lip of the blastopore expresses Noggin. It also expresses a gene called cordon, beta-catenin, siamois, and goosecoid. Remember, the dorsal lip is where future endoderm and mesoderm cells migrate into the embryo. One of the important interactions that have been shown is the interaction between endoderm cells and the overlying mesoderm cells. The interaction between the endoderm and mesoderm cells is critical for mesoderm cells gaining their identity and for the mesoderm cells forming somites and muscle and bone accordingly, later in organogenesis. This mass migration continues to occur through gastrulation, as you've already described in the introduction to your lab report. Cells pass by each other and induce each other to form the 3 germ layers, so that at the end of gastrulation, we have a fully formed ectoderm, mesoderm in the middle, and endoderm on the inside, with a space on the interior of the embryo called the arc Entraron. So here we can see in the 27 to 29 hour chick embryo, we're looking down on the chick. Anterior is to the left and the most posterior is to the right. Now in the anterior-most part of the embryo, which I've got cross-sectioned here, we can see that the neural groove is bending in and bringing ectoderm up and around. Now if we move slightly posterior, we see that it's folded less. Now what's going to happen is this ectoderm is going to make a new tube that is then going to be the neural tube, which is going to become the forebrain, the midbrain, and the hindbrain, as well as the rhombomeres on the dorsal side of our chick. Now, the genes that are expressed in the anterior-most head position that cause the formation of the forebrain are Noggin, cordon, and follistatin. That's right, the organizer is responsible again or the genes responsible for the organizer are responsible again for forming the brain, specifically the forebrain, which is responsible for controlling the organism. Now we can see here that mesoderm is going to be pinching in and creating somites on either side of the notochord. Now, the notochord is a fluid-filled structure that's transient in this embryo and will not be there when it's an adult, that signals ectoderm above it to construct things, as well as mesoderm to either side. Finally, the gut tube is here on the bottom. So the neural fold continues bringing dorsal ectoderm up until it makes a neural tube and then has ectoderm lying on top of it, as we can see in this diagram. Now, that neural tube is going to form something called the telencephalon, most anteriorly, because of the expression of genes DLX, OTX1, and OTX2, as well as chordin and noggin and follistatin. The midbrain is going to form, or what is called the diencephalon before it's called the midbrain, from the expression of OTX1 and OTX2. And then the hindbrain is going to form where engrailed is expressed. Finally, further down the brain stem, rhombomeres will be made, which will innervate the front of the face, and Hox genes 1 through 3 are going to direct the formation of those rhombomeres. Now, we see here another cross-section of the embryo with a fully formed neural tube. I've left a space here at the endoderm to indicate the anterior intestinal portal will form here, though, of course, the embryo would be turned sideways at this point, and I have it facing down towards the yolk. The anterior intestinal portal is what we call the mouth at this point. It's not a mouth yet. Now, the notochord runs through kind of the center of the embryo, and to either side of it, mesoderm is pinching into these chunks called somites. They're going to form vertebrae and body muscle, as well as some other things. They're going to form somites because of signals from the notochord and the neural tube. Now, right here we have what's called the fourth germ layer. Between the top ectoderm and the neural tube, cells are going to break off, driven by the expression of genes like Wnt1 and snail, as well as other things. These cells in the face, when they break off, are going to move around to the face of the embryo, and they're going to form rostral skull structures, like the mouth and the beak of the bird, and several other things. However, these cells will migrate from areas all the way down the embryo as development proceeds from anterior to posterior, and these neural crest cells are going to make things like pigmented skin and feathers in the bird. In addition, here in the head, we're going to have some expression by OTX1 and OTX2 that's going to cause this lateral protuberance to grow off the diencephalatic wall of the neural tube. That's the hindbrain, actually, right? Or right there at the midbrain and the hindbrain, okay? And PAX2 and PAX6 expression is also going to be involved in causing this protuberance to grow and meet the lens placode that is going to develop from the overlying ectoderm because of PAX2 and PAX6 expression. That's going to make the embryo's eye. And now here we see the entire embryo. We see snail initiating neural crest migrations. We see the bronchial arches forming in the heart. And we see that Hox genes 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13 are expressed from anterior to posterior down the embryo's body. And as we know, the apical ectodermal ridge of the forelimb is going to form here on the anterior-most boundary of HOX6 gene expression, right? And that's going to initiate FGF4 gene expression, which will initiate sonic hedgehog gene expression, and that will develop the posterior structures of the forelimb as it grows out from the embryo. And the hind limb bud is going to be forming here around the highest concentration of HOX9 gene expression.